Good evening, everyone. Hope my voice is audible. Give a thumbs up if I'm audible. Okay. Welcome to today's webinar in Bioassay, which will be taken by Dr. Padmanabh. Sir, he is an associate professor in one of the reputed college, and he has been mentoring many persons, many students as well, and he has been our one of the most eminent teacher. And yes, this is a brief introduction about Dr. Padmanabh sir. Yes, over to you, Padmanabh sir.
Good evening, sir. Thank you for the introduction. Can we continue, sir, or should we?
Yes, you can start, sir.
Okay. Thank you, sir. My screen visible, sir?
Yes, it is visible.
Okay. Thank you, sir. Good evening to all. So, welcome to this bioassay webinar session today. Myself, Dr. Padmanabha TS. So, in today's we'll go through briefly how bioassay helps in terms of drug development. So, bioassay a tool in preclinical drug development. As you know that preclinical means just before going to clinical trial, before handle any of the substance or the molecules in human beings, we're going to test those molecules in the animals. Or in terms of seminars, we will be testing molecules in vitro studies where we will be using the cell as well as the tissue samples from the animals. So coming to the overview of this session, so this session will be dealt through these headings. So principles of bioassay. So what are the applications of bioassay? So how do we identify the bioassay into, and what are the methodology we follow in conducting the bioassay? And also we'll learn about how this bioassay will be conducted in short. So if you have any doubts, you can leave it in the chat box, or you'll be free to ask me any questions at the end of this session. So coming to the basic history of bioassay, as it was originated late in the 18th century, and as you know that Paul Ehrlich, so he was the one who gave a insight towards the bioassay. He was the one who initiated the standardization of diphtheria toxin. From then onwards, it became a common method for standardizing the substance via biological system before it goes to the clinical trials. So what do we understand by bioassay? So bioassay, when we divide it into two words, bio means it is a living material. So it's a living thing. Assay means we are going to assess the tissue with respect to the molecules which we are choosing. So basically it is an assessment of potency of the substance using a living system. So here the living system means it can be a in vivo study as well as the in vitro study. So basically why we want to do this bioassay, it helps to determine the effect of unknown natural substance. To know that when we derive any of the substance from the natural product, we will not be knowing how it is going to affect our biological system. So before giving a trial on humans, we want to check these effects on the animals as well as the in cell tissue. That is where the bioassay comes into place. So we are testing a natural substances, that is unknown natural substances, to determine their effect, means how they are potent, how efficacious they are in terms of producing the effects on the living system. So please remember, bio means we are dealing with the living system. So coming to the definition of bioassay, So basically, it is defined as a comparative assessment of a test compound versus the standard compound. So here the test compound means it is the unknown compound which we are getting extracted from the natural substance. So standard compound will be a standard one which will be comparing the test compound with the standard compound which is already existing in the market. So basically we conduct in the living animal or the biological tissues. So what we are going to determine by doing this bioassay, we're going to arrive at the dose response relationship. So by knowing the dose response relationship, we can compare how effective is the test compound with the standard compound which is available. So coming to the types of bioassay, we have two types of bioassay broadly. So it can be classified into quantitative bioassay, where it measures the DRS, that is the dose response curve relationship. It can be a direct type or an indirect type. Whereas the other type of bioassay is a qualitative type of bioassay. In qualitative type, we're going to observe only the physical effects, like what are the deformity, what are the defects which the unknown substance can cause. Mainly, it is used when effects cannot be quantified. So here bioassay mainly what we deal here will be of more of quantitative bioassay where we'll get a dose response relationship. So coming to the methods of drug assessment. So usually when we assess any drug, so these are the standard formats which we use in the assessment of the drug nature. One is the physical method, second one is the chemical method, and third is the biological method, that is the bioassay. So when we compare these three methods, all these individual methods has got the advantages as well as limitations. So in this session, we'll mainly concentrate on the bioassay, that is biological method. So what is the advantage of bioassay? Why we need to learn about the bioassay? As we know that nowadays the animal studies are banned. Unless we test for any new compound or with permission, we can do this bioassay when we are going for any preclinical trials. So it gives us improved reliability with respect to the effects of the unknown compound. It is highly specific. For example, when we give any drug to drug on a living system, or when we extract some substance from the natural product, we want to see for how specifically it responds with the individual tissue or the individual organ. So bioassay is very highly specific in nature. It also gives an accurate measure of the response, as well as it is very sensitive to the individual molecule that we are extracting. And also it gives a better probability in terms of detecting the effects, since we are comparing with the standard drug or a standard dose of the known molecule. So we'll be able to easily compare how this unknown product is producing its effect. And also at preferred one, specificity is crucial. So we need to have a specificity towards the individual compound and how this individual compound will respond to the individual tissues as well. So whenever we require a specificity, we go for a bioassay. So when we compare with the chemical assay, so usually chemical assay is easy to standardize when compared to the bioassay. So bioassay gives a greater specificity, as we discussed in the earlier slide, but whereas the chemical assay gives the greater precision. So if we take an example of when we expose any living tissue or living thing to any of the antibiotics, so what we are going to see when we expose the tissue to the antibiotic means whether the growth is inhibited in the tissue or not when we expose to the antibiotic. That specific we are seeing. So whether that antibiotic is specific to that organism, whether that antibiotic is inhibiting that growth of the organism, etc. So that tells about the specificity. So bioassay gives us the greater specificity, whereas the chemical assay gives us the greater precision. In simple terms, when we look at the any water pollution or any air pollution, we look at the what is the substance which is present in the air which is causing the pollution. So in that terms, chemical assessment provides a very precision amount or the precision number of substances or the particulate which is present in the air or the water. So please remember, bioassay gives us the greater specificity, whereas chemical assay gives us the greater precision. So when we integrate these both of this bioassay and chemical assay, what we are going to achieve is we are improving the reliability as well as we are reducing the variable response with respect to the tissue. In turn, finally, we're going to improve the reliability so that it can be measured many number of times. So coming to the substance which are used mainly in the bioassay. So mainly, as we told, we use a substance which are mainly derived from the natural product. It can be from a plant source as well as the animal source. And this chemical or synthetic drugs are usually excluded because we know the structure as well as how this structural change can bring the effect. So already chemical standardized methods are available and it's easy to predict the effect when we talk about the chemical and synthetic drugs. So when we talk about the biological product with respect to the tissues or the product which are derived or extracted from the plant or the animal source, and we don't know how those active molecules will going to affect the living system. So that's how the bioassay will be the different from the chemical as well as the synthetic or chemical assay. So objective or the purpose of bioassay. So what is the objective of bioassay? So bioassay consists of three main components. So it deals with mainly stimulus. So we give a stimulus. Subject will be the tissue. Based on the stimulus, the subject is here. The subject means the tissue or the cell line which we are using, and the based on the stimulus which we give to the tissue, we're going to have a response. So these are three things, three main components which we need to see under bioassay. So basically it includes an objective where we're going to identify various unknown compounds. We quantify the unknown compounds during this screening procedure. So usually we are screening how effective at what dose or what potency the drug is producing the effect, whether it has got low potency, whether it has got high potency, or what are the toxic limitations, etc., can be studied with respect to the bioassay in comparison with the standard molecule. So one example is whenever we go for a commercial production of the antibiotic, initially we have to go for the bioassay where, as we saw in the earlier slide, we check for the specificity regarding example which was taken was whether that antibiotic will going to inhibit the growth of the bacteria or not. In such a way, we can quantify the drug unknown drug into the minimum inhibitory concentration as well. So what is the minimum effective dose and what is the lethal dose, etc., can be made out through the bioassay. So coming to the principles of bioassay. So how does this bioassay will going to function? So basically, it's a quantitative procedure. Please remember, it's not a qualitative procedure, it's a quantitative procedure. Basically, we use a functional response. So how the living system will going to respond to the stimulus. Here stimulus means the unknown compound will be a stimulus where the living tissue will be the subject, and by giving the unknown substance onto the living system, we're going to see how that living system will going to respond to the subject respond to the unknown product. So it can be of in vivo or in vitro. So it compares the potency rather than the direct responses. So basically, what is the potency means here? It tells about at what amount of drug will get such response. So we need to do a serial dilution of various concentration. So based on that concentration, we're going to measure the response with respect to the living system. basically it determines the amount of substance. For example, so whether when we take a clear example of the when you take a aspirin, we know that aspirin has got different effects at different doses. So how we quantify it based on the dose response relationship. So at lower dose it has got antiplatelet action, at higher dose it has got antipyretic action, as well as higher doses it has got analgesic action with weak anti-inflammatory action. So such can be quantified through the bioassay based on the response. So it basically measures the relative activity or potency, a quantitative measure of biological activity. And also you should remember that potency increases with higher concentration of the substance. That means when we give a higher concentration, the potency will increase. Means if the concentration is high, at the lower doses itself, it will be able to produce the response as well. So the effective bioassay will yield a dose response data within a broad and practical concentration range. So what we are going to analyzing when we get a dose response data through the bioassay, we're going to analyze what is the range of drugs which we will be able to uh, assess. What is the minimum effective dose, what is the maximum effective dose, what is the lethal dose, and also we can come at the mean concentration at which the drugs will be more effective. So apart from that, we also need to check about the errors which can happen in the bioassay. As you know that errors will happen in any of the assessment. So does in case of bioassay as well. So one is the response variability. The bioassay may vary with respect to the response, whether it can decrease or increase based on the procedural errors. So even a minute error will be a minute variation. It will bring about the much larger extent of variability will not be able to quantify. So the preparation of the bioassay as well as preparation of the components of bioassay is very crucial in terms of getting accurate results. So what are the types of error we can expect, and when we think about these errors, we need to try to minimize these errors while doing the bioassay. So we have a biological variation. So this is very important. So there can be a natural difference existing between the individuals as well as animals. So even though you collect a tissue from the same species of animals, those tissue can respond differently. And typically results will be in a symmetric frequency distribution, but it can vary based on the individual responses as well. So what we need to do, how to correct this biological variation, we need to use a standardized tissue and maintain the consistent procedural values as well as the procedural steps. That standard operating procedure with respect to the tissue has to be done to avoid these biological variations. And also when you are selecting the animals, also you need to select a specific age group of animals where the animals tissue will be responding to your unknown product. So otherwise also there can be more biological variations. Coming to the second variation, apart from biological errors, you can also expect the methodological errors. So it is of human errors. So mistakes will happen during tissue selection when you are preparing a tissue for mounting on a organ bath to see the response, and also when you are recording the responses when you mount the tissue in the organ bath. So every steps is very crucial when we if we want to get accurate results, or when we want to know the concentration at which these unknown substances are producing the results. Apart from that, we also have an experimental error where faulty apparatus. So we need to check very carefully the apparatus setting. So calibration is very important. Even a minor issue with respect to the calibration can be very difficult in measuring the or quantifying the amount of unknown substances producing the responses. So what are the common issues and corrections which we need to take care? So one is the non-standardized procedure. So we need to standardize all the procedure beforehand. The instrumental issue has to be checked. We need to calibrate each of the instrument properly, and poor tissue handling. So make sure that when you are collecting the tissue and preparing the tissue, it should be minimally handled, so that especially during cleaning process. So we need to minimize the excessive cleaning. So if you handle more, the tissue will not be sensitive as well. And incorrect physiological salt solution preparation. So we need to accurately measure each of the salt or the electrolytes when we are preparing the physiological salt solution, so that we can maintain the correct pH for the tissue. And drug dilution error. So mixing has to be done very slowly, and it should be in a methodological manner, and avoid vigorous shaking. So again, these things will going to affect the tissue response. And down regulation and sensitivity loss. So if you're when you started with the bioassay, and tissue would have been mounted, and when you're giving interval between the responses, you'll be doing a washing. So during that, please avoid the overwashing as well. And if the tissue is not sensitive, you need to go for a newer tissue to get a response. Animal variability. So you need to use the same species, same gender, the same status of the health with respect to getting the tissue response from the animals. And lab conditions should be constant throughout the experiment. So, and as well as improper handling, ensure the trained personnel should be involved with respect when we are doing all this experiment. So these are the common errors which we encounter during bioassay, and this can be handled with gentle care, as well as if we have this prior uh, 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 what has to be done before the procedure, during the procedure, etc., will be able to minimize these errors in the bioassay. So where we can apply this bioassay? When we are done with the bioassay, what is the end result of this bioassay? So it can be used when chemical methods are unavailable for estimation of the potency or the amount of the drug. So you can standardize the natural origin drugs which are derived from the plants and animals with unpredictable structure. So usually when the molecule is extracted, will not be knowing the structure as well. What is the chemical structure? So unless we know the chemical structure, will not be able to go for chemical assay as well. So in such conditions, you need to go for the bioassay to quantify what are the active ingredient, at what amount or the what potency or what dose this active molecule derived from the plants and the animals are effective. And it can be used, it is used for screening a newer compounds for seeing the biological activity, while quantifying the biologically active substance as discussed. So especially when we are using histamine, acetylcholine, serotonin, adrenaline, bradykinin substance, P prostaglandins, we make use of the bioassay. And also it helps in determining the safety parameters, that is the effective dose can be determined, toxic dose can be determined, lethal dose can be determined. So all this are the applications where we can get it from doing a bioassay. So before going to the methodology, how individual bioassay can be done, this is the structure of bioassay, where you can see here the one is the power input, this is the switchboard, and this is the eating rod. So basically you will have a Stirling's a rotating drum, chymograph, and this is the organ bath. So usually there are commonly used or the single organ bath as well as double organ bath, and industrial side you will have a multiple organ bath as well. So this is the eating electrode, and this is the thermostat, and this is the coiled physiological salt inlet to the inner bath. And next is the outer bath is there. This is the outer bath, and here you have a what is called as the this is the fulcrum, and this is the tissue where you will going to mount it. This is the inner bath of the physiological salt solution, and you have a thermometer to regulate the temperature, and you have a recording lever. So this is the fulcrum. So fulcrum is very important in terms of adjusting the magnification for an individual tissue. And below the fulcrum you have a hanging thread where you need to tie it with the tissue here. So into this inner bath you're going to administering the unknown substance solution into this inner organ bath, where you will be having a tissue samples. So this is a fulcrum, and you have a recording lever. So this recording lever is adjusted to the fulcrum, and this will be your stylus, and this will be writing on the chymogram drum. So drum will be rotating. So there are adjustments which can be set for rotation, and this Sterling's rotating drum is there, and you have some levers and gears to adjust the mounting of the chymogram sheet and to record the responses through the lever which is attached here. So whenever you put any unknown substance to this, the tissue, whenever the tissue is sensitive, the tissue will respond, and those responses, when the tissue contract, the same contraction will be recorded in terms of response here through the lever. So once the phase of the response is over, you need to give a cycle break, and wash has to be done, and again you can go with the response with respect to the other concentrations as well, to check at what concentration the drug is more effective, and it is we can compare with the standard drug whether it is producing a same response as to that of the standard drug, or whether it is much more effective than the standard drug or lesser effective than the standard drugs. So this is the common thing, how it looks like. This is the organ bath, and this is the Sterling's rotating drum and a chymograph, and this is the physiological salt solution where you will be preparing, and you will be loading here. From this, the physiological salts will be moving in. So this is a simple diagram which shows about the how the setup of the organ bath and chymogram and Sterling's drum are made out, and also along with that, this tissue, to maintain the tissue viability, you need to aerate with the oxygen supply. There will be oxygen aerator as well. You need to set the oxygen aerator so that tissue is viable till the experiment ends. And next, this is the just a diagram showing this is the single unit organ bath. You can see here the inner organ bath is there. To this, the tissue will be kept here. There will be this is the fulcrum. Here you can see the lever, and this is the writing part which will be in contact with the chymogram drum. And here you can see a double unit organ bath. The two tissue can be kept here, and you can record the two tissue responses at a time. So this is the thermometer where it will maintain, sorry, this is the heating coil. So it will maintain the temperature. And apart from that, so this is how the organ bath looks like. And next, coming to the organ bath setup. So traditionally, organ bath is commonly used in academic setting. So these are the component as you saw in the earlier setup: inner organ bath, water bath coiled with inlet for physiological salt solution, stirrer, heating coil, chymogram with revolving motor. So this is the setup in academic setting, whereas industrial setting, there is advanced versions as well. They have multiple organ baths with greater precision and automation. So individually, we need to, in academic setting, we need to set up all those things, whereas in industrial settings, all those things will be automated. So when we talk about this organ bath was first used by the Rudolf Magnus in early of the 19th century. Initially is standardized with respect to the intestinal strips. So this was the basic design in terms of getting a response from the tissue with respect to the unknown substances. So Rudolf Magnus has a greater contribution to the bioassay. So what are the essential parts and uses of the individual organ bath? So there we know the types of organ baths. As we saw in the earlier picture, we have a single unit organ bath, which was developed by the Rudolf Magnus. So it contains a single unit organ bath. So inside you have a single unit organ bath where single tissue can be mounted and check for the response. So commonly used in academic labs. Whereas the other type of organ bath is a double or multiple unit organ bath. And this has been initiated and introduced by the Gadam, the scientist Gadam. Here it will have a inner organ bath, will have two or more inner organ baths. Simultaneously or parallelly, you can test many of the tissues as well. And automated multiple units will be used in industry. So when compared to the multiple single unit organ bath, multiple organ bath will be automated, and it gives a more efficient responses as well as the result in terms of drug discovery. So in industrial side, they use the multiple unit organ baths. So there is a rotating drum and chymogram. So it is a Sherrington drum which records the muscle response. So usually the speed will be adjusted for a tissue type. Standard rotation will be 1 revolution per 96 minutes. And chymogram paper, which is a smooth side will be mounted outside, and rough side will be mounted inside onto the chymogram drum. Then we have a outer pocket or outer water jacket which surrounds the inner bath to maintain the temperature. Usually it is made up of glass or steel. As you can saw in the earlier slide, it was a glass organ bath. The inner organ bath will also be a transparent. It is usually made up of glass. It has a capacity ranging from 5 to 50 ml. So usually it is used to observe the tissue response. So whenever, if you have done an experiment, you can see the very beautiful thing on the how the tissue will be contracting when you are dealing with the any ileum, rat ileum, or the mice, or the guinea pig ileum, or even if you take a chicken intestine. So you can see a beautiful response with respect to how it contracts and how the contractions will be reflected in terms of the response on the chymogram paper. And we have a tissue holder and an oxygen supply. This will be used for to secure the tissue to maintain the viability we'll be using. Then we have fulcrum. Usually it supports the liver for vertical movement. So this will take a responses of the tissues. Then we have a heating coil and thermometer to maintain the temperature. We have a stirrer to make a uniform temperature distribution. Then we have a fixing of record. So mainly we use a fixing solution which is made up of Shella and colophony in methanol. This will be kept for 5 to 6 days, and it will we'll get a finer collection without any debris later after 4 to 5 days. And same solution can be used to mount or fix the recording which was happened on the chymogram. So usually it is used to preserve the tracings of the recording, that is the tissue response recording on the chymogram. So you need to mount as well as to fix it. So otherwise what happens, it will be gone. So this is the complete what are the components you saw. Now what you need to know is what is the steps in terms of conducting the bioassay. So basically we need to check the instrumental calibration, all errors with respect to the human error, biological tissue error, as well as the mechanical error with respect to setting up of organ bath should be minimized as much as possible. So first we need to complete the adjustment of the organ bath, adjust the liver and the balance, very, very important. So this takes very precision, and when we are doing with the single unit organ bath, when we are doing it takes a lot of patience and lot of time to adjust the liver and balance. So even if you don't adjust the liver and balance it properly, even if the tissue is responding, you will not get a proper recordings on the chymogram. So basically that very important step, that is the adjusting of the liver. Then the animal and tissue selection, you need to check which tissue has to be selected when you are testing a unknown compound. So tissue has to be selected, and before once you have thought that the tissue has to be selected, you need to go for surgical procedure to collect the tissues, abiding to the particular ethics committee regulations and the national guidelines has to be kept in place before conducting all those surgical procedures and the any animal experiments, and collection of the appropriate tissue and keep in the physiological salt solution. Why we need to keep in physiological salt solution? This physiological salt solution will maintain the environment similar to that of the particular animal uh, 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 homeostatic environment will be maintained. So then the tissue is attached to the organ bath. So first you have to keep, once you remove that, it has to be kept in the physiological salt solution. So you maintain the viability of the tissue solution. If you don't keep in the physiological salt solution, the tissue viability will be gone and the sensitivity will be lost. So once you select the tissue has to be attached to the organ bath, that is the inner organ bath, and you give a time for relaxation. So it's a relaxation time to the tissue, 30 minutes or 45 minutes, etc., based on the experiments which we are conducting. And meantime, prepare a standard solution. That is, a serial dilution has to be done to maintain the concentration. So both serial dilution has to be done for standard solution as well as the unknown compound. And based on the standard responses, we will be calculating the concentration of the unknown compound as well. So after the relaxation test, any concentration of drugs for response. So preferably high concentration dilution has to be checked initially to check for whether the tissue is responding or not. Then you go, once you check with the high concentration, then you go for a standardization. So then you standardize the tissue response with the same dose. You need to take a subsequent 2 responses, and drug concentration is selected based on the trial and error methods. So select the lowest possible measurable response. So basically all this bioassay is about you're comparing with the standardized drug. So you need to standardize the tissue response of the unknown concentration which you are taking with the known concentration of the standard drug which you will be utilizing to compare the concentration of the unknown compound. So you need to prepare a dose relationship curve for standard drug initially, and prepare a test drug dilution again, and then make a dose response curve with respect to the test drug. And following that, you need to select the assay method. So whether we are going for the bracketing assay, matching assay, or interpolation, or a 3-point or a 4-point assay, based on the what we want to see, whether we want to see for toxicity, we want to see for the potency or comparison of the effective dose or the maximal lethal dose, etc. Based on that, we need to select the assay, and also based on the volume of tissue which you are going to select the sample, also we are going to select the assay method. After taking several responses, what you need to do is that you need to measure the response. How are you going to measure the response? We are going to measure the height of the each responses, and you go for a calculation. So basically we need to do the calculation with respect to the height which we are taking from the responses. So we need to compare with the standard responses, and again it's a complicated this thing where you need to go with the graphical representation. You need to calculate the log of the drug and anti-log of the drug to arrive at the concentration of the unknown drug which is more effective at what concentration, etc. So this is how the tray is maintained, where this is the tray used for fixing solution, where the fixing solution is made of shellac in methanol, and you need to allow this solution for 5 to 7 days so that it can settle down. So all the particles will be settled down at the bottom, and you need to filter, and you need to use this solution. So this solution, and this is the paper clip, is the chymogram recording. This will be attached to the chymogram when you are recording. So once the experiment is done, you remove it, and you need to So here you can see the responses. So these responses has to be fixed using the shellac in methanol solution. Then coming to the dose response relationship. So basically, what is the dose response relationship? It tells us about the potency comparison between the standardized drug as well as the test drug. So what is the dose response curve? Basically, it is nothing but the at what concentration we are getting the responses. So we are going to plot this in an ascending order, and we are comparing these plotted graphs with respect to the standard drugs and the unknown compound. So usually what we need to do is we need to avoid prolonged ascending sequences. So prolonged higher dose administration can decrease the tissue sensitivity as well. So to maintain the tissue sensitivity, what we need to do is we need to go for intermittent dosing as well as washout period has to be given so that the tissue availability will be maintained. And next is the curve characteristics. So we need to check whether we are doing the experiment correctly or not. So to measure this, we need to check for a linear relationship exists or not with respect to the log dose and the response. So this has to be done after conducting the experiment, where you need to measure the length or height of the responses from the chymogram, and you need to plot on the graph, and you need to check for linear relationship exists or not. So usually what happens is the parallel DRC of the test and standard indicates a valid potency comparison. If there is no parallel dose or the drug dose response relationship is not seen, then we need to think twice about the validity of the potency comparison. So that's what the non-parallel line suggests the errors in the methods. So usually we need to make sure that if we are followed a particular steps according to the standard operating procedure, we'll be able to get at least a linear relationship with respect to the standard drug and the test drug, so that the parallel DRC should be maintained, so that only that time we can tell this drug has achieved the valid potency comparison. Then we can go with the regression analysis to assess the potency correlation. So this can also be done. So this is the diagrammatic representation where you can see the ascending dose response plot. So as you can see here, there is a standardization of the tissue response. So as we discussed in the steps of how to conduct a bioassay, first we need to standardize the tissue response. So these are the lowest dose or the lowest concentration. Then you standardize. So you are giving a lowest concentration. So same concentration at twice. So you need to check for whether it is standardized or not. Later, you can increase the concentration, and you can see the response. There was a linear ascending dose response, and also later, once it achieved the maximum height, you can check with the higher concentration whether it is responding or only already it has reached the maximal dose response. So you can also see the ceiling effect. So once you have reached here, again, if you give the 0.8 concentration of this concentration, what is happening is there is a ceiling effect or the supramaximal effect. Again, to check for the tissue sensitivity, what you can do is repeat the initial doses so that it will produce the initial response. If it is producing the initial response, that means to say that even still the tissue is viable to the experiment. And this is the intermittent order dose response plot, where initially you need to standardize the tissue response initially, then go with the higher concentration, and intervally you need to check with respect to the higher and the lower doses. So you can see 0.4, then you go with the lower dose of the higher concentration, 0.2, 0.4, 0.8, and check for the maximal dose response and what will be the ceiling height or the supramaximal response with respect to the dose. And to check for the viability, you check with the minimal concentration. So this is the response what you get, and this is the baseline. So from the baseline, you need to measure the height of these responses on the graph. So this is a linear parallel relationship. That is the graph A shows that this is the valid potency for comparison of the test drug and the standardized drug. whereas there is a violation or there is an incorrect linear relationship exists between the B graph. And whenever you're choosing the dose, please make sure that you need to select the dose from the 25 between the 25% or 75% of the response. So that should not be a maximal dose or the lowest dose. The dose has to be selected within this range, that is 25 to 75% of the linear response range. So this is where the drug will be able to respond to the unknown tissue. So unknown tissue will going to respond response good at these concentration between 25 to 75% response. Then next we have a physiological salt solution. Please make sure that we need to prepare this each of this fresh solution has to be prepared, and each of this concentration, if there is a difference with respect to any minute in terms of weight when we are calculating and when we are adding in preparation of the physiological salt solution, it makes a big difference, and you're going to see a variability in the response. So all this concentration should be exactly measured, and we need to do the accuracy in terms of preparation of the physiological salt solution. So if you look at the bioassay study, you can say the heart of the bioassay will be the how good you are going to prepare a physiological salt solution. So physiological salt solution is the main component where it will going to serve, it will going to make tissue viable for a longer period of time. Unless you prepare a salt solution properly, tissue viability will be lost. As you can see that the common ingredients used for the salt solution prepare is sodium chloride, sodium bicarbonate, the glucose, potassium dihydrophosphate, sodium dihydrophosphate, potassium chloride, magnesium sulfate, magnesium chloride, sucrose, calcium chloride, and aeration. All these things have an individual importance, and most commonly used solution is the thyroid solution. So based on the tissue, you're going to prepare the particular specific physiological salts so that the tissue will be able to maintain. So all these solutions are prepared basically to maintain the tissue similar to that of the how tissue was there inside those animals before. So basically it mimics the internal environment to sustain the tissue outside the body. The commonly used solutions will be a ringer solution and the thyroid solution. So the main components will be the ions, and energy source will come from the glucose. So usually what you need to look is, usually it is prepared using a distilled or double distilled or deionized water, and basically you need to add some of the step has to be prepared very carefully, that if you are going to add calcium chloride in the beginning itself, what it will going to precipitate with bicarbonate. To avoid it, you need to put the calcium chloride in the last. And use of carbogen, that is 95% oxygen and 5% oxygen for aeration to prevent the alkalinity. So this has to be maintained. Aeration has to be maintained properly. And tissue specific selection. So thyroid solution is selected for innervated muscles, and Krebs solution is selected for the nerve muscle preparation. Usually you need to maintain the pH at 7.3 to 7.4. And regarding storage, you need to always prefer using a fresh preparation, maximum 24 hours as it can be kept in place. If longer storage has to be if longer storage it happens, then you need to give away the calcium and glucose until the use. At the time of use, you need to add a calcium and glucose. So precautions: maximum error in composition. How much you need to have a error? Less than 1%. That's what I told. It is the heart of the bioassay, where the if the physiological salt solution, if you have prepared correctly, then the off work of bioassay is done. So that's why the error is very, very minimal here. Error should be very, very minimal. So this is how the physiological salt solution are made, and these are the components and their utility, where basically it maintains the similar homostatic the environment as to that of the tissue which was staying in the living tissue before extracting it. So all these are the roles of each of these components. So basically, the bicarbonates will maintain the pH, and magnesium's chloride will support the tissue activity, which will reduce the spontaneous tissue reactions, and it also supports the neurotransmission. The glucose, in other hand, it provides a nutrient to the tissue in case of in vitro conditions. And these are the important points with respect to some of the individual physiological salt solution. So as we discussed, thyroid solution is used for mammalian soft muscle. Frog ringer solution ideal for amphibian tissues. Ringer lock solution is used for heart muscle preparation. Krebs solution is commonly used. It can be used for any of the tissue. So Krebs solution has got homogeneity or homeostatic environment in all of the any of the animal tissue which you take. The Krebs cycle, the Krebs solution will be able to maintain the tissue viability. And coming to the liver and magnification. So as you know that, very important, it takes time. The patience has to be there to in terms of balancing the magnification as well as liver. So you need to balance the tissue as well as the liver, so that the what are the response we are getting at the tissue level, that has to be recorded on the stylus or the at the end of the liver on the chymogram sheet. This is how basically it principles. The static equilibrium, basically it's maintained through load force into load arm, which is equal to effort force into effort arm. So what are the components of liver? The effort arm is the where force is applied. The load arm is the where the effect is absorbed. So where the effect is absorbed, and effort arm where the force is applied. Fulcrum will be the pivotal point. So there are types of liver. The commonly used will be the type 1 liver. It is usually used for recording and amplifying the tissue response. So if you are not properly magnifying the liver, if you are not properly keeping the fulcrum at particular point for that experiment, the amplification will not occur, and the graph which you get will be of minimal height and will not be able to compare with the standard as well as the test drug cannot be compared. So to have a proper magnification, the liver and the fulcrum, that the effort arm, load arm should be properly faced. So the stylus. So what is stylus? It's a contact point on the chymogram where that will going to record the responses. Usually it will be made from the steel or a aluminum or a wood, or sometimes you will be using X-ray sheet as well, or a hard plastic copy. Magnification rule: when you are using a slow contracting tissue, the magnification should be of 10 into 15, whereas fast contracting tissue, it should be of 5 to 10 magnification. So these are the type 1 liver, type 2, type 3, and here you can see that this is the fulcrum, and this is the load, and this is the effect. So what will be the load? Load will be example, if you are choosing a tissue, this will be the tissue end, and this will be the effect end where you will going to record the response. So this is how you will going to set up a tissue in on the unit of organ bath, so that fulcrum, effect arm, and load arm is adjusted. Only if this is correct, you'll get a proper magnification. Later you'll be able to compare the test and the standard drug. Unless if you are not if this goes wrong in magnifying and liver adjustment, what will happen is, even though you get a tissue load here after administering the unknown compound, the effect will not be recorded properly. So magnification and its adjustment. So what are the types of transducer we need to understand? We have isotonic and isometric. The isotonic records the length chain, not the tension. Example is a simple liver and the frontal liver, whereas isometric will going to record the tension chain but not the length. So with respect to whenever we get a muscle twitch, we talk about measuring the tension change. So it requires special equipment, whereas isotonic for recording the length change, that is the contraction, etc., we can use a simple lever or a frontal writing lever. Coming to the isotonic setup, so magnification will be a key for amplifying the response. As we discussed earlier, for slow contracting tissue, we need a higher magnification. For fast contracting tissue, we require a 5 to 10, whereas here we require 10 to 15 magnification. The magnification formula, how we will calculate it, it is the distance from fulcrum to the writing point is A, and distance from fulcrum to the tight tissue is B. So divide the distance when we'll get a magnification value. So that is the length of the effort arm, length of the load arm. So effort arm will be the writing point where stylus will be in contact with the chymogram, and tight tissue will be at the end of the where the inner organ bath will be holding the tissue. That will be the load arm. So this will be the stylus. This is the writing point. So this distance is called the effort arm, and fulcrum to the tissue where it is tied, the B will be called as a load arm. So A is the effort arm and B is the load arm. So when we divide it, we'll get a magnification. So we need to check for a magnification before conducting the experiment. So these are the other types of lever which we need to know. So I'll not go into detail, just for a glance. So remember, we have a simple lever, gimbal lever, oxonotonic lever, we have a frontal writing lever, we have a sprung lever, we have a torsion lever. So usually many times we use a simple lever which is made up of stainless steel or aluminum or a wood with stylus which will be attached to it. So this is the string and this will be the stylus. This tip will be placed onto the chymograph drum. And this is the Starling heart lever which will be used in the experiment. So coming to the dose response relationship, the principle which is used behind dose cycle and the responses. So based on the dose response relationship of the test and the standardized drugs, we need to go for how many dose cycles has to be there, and what is the frequency at which we need to administer the drug, and at what rate we're going to get the response. So basic requirement, tissue must be always relaxed, otherwise you will not get a specific effect of unknown compound on the tissue. Proper load for specified time has to be there. Baseline recording is crucial for uniform response. So you need to record the baseline properly to check whether the tissue is responding or not. So when you go for a higher concentration, the tissue may not respond at all, and you may not get the response with subsequent concentration. So basically, you need to check for the baseline recording first and make sure the tissue is viable, and also make sure that it is aerated properly and physiological salt solution is maintained at proper temperature as well. All these things will going to matter in terms of getting the response from the tissue. And biological variability, what we can expect is the plus or minus 10% variability is accepted. More than that, the acceptability will be lost. So tachyphylaxis, there is a decreased response after repeated dose, and also the dosing frequency or dosing interval between the first dose and second dose or a subsequent dose should be planned in such a way that there should not be in a quick hurry. So what happens? Tachyphylaxis. So there can be a down regulation of the receptor, or tissue sensitivity will go down, so that the unknown drug may not respond as well. So when we keep on administering the unknown compound on a quick succession of time. So what are the three phases we need to remember in terms of base cycle? We need to record the baseline recording. Then after recording the baseline recording, we need to record the dose response. So once we have recorded with the dose response, then we need to give a washout time, or a washing of the tissue has to be done, or a washout time has to be given before injecting the second concentration. So there should be a gap between the first drug administration to the second concentration drug administration, so that we'll be able to get a response from the tissue. Otherwise, what will happen? Subsequently, if you are not washing the tissue will have that concentration and it may not respond as well due to tachyphylaxis. So dose cycle 3 minutes for fast contracting tissue and 5 minutes for slow contracting tissue, and include at least 2 tissue washes in between so that there will not be concentration of or the concentration accumulation will not happen, so that you can make tissue viable for a longer period of time, so that desensitization can be removed. So you can make tissue much more sensitive when we include a tissue washes in between. So this is how, what are the 3 responses what we saw. So this should be a baseline. Then we need to check for a dose response, and this is for slow contracting tissue, and this is for fast contracting tissue, and this is the baseline for next dose. So here you can see that A is the dose cycle from initial after baseline. So you'll get a baseline before injecting, you inject the drug, so then you will get a response, you stay for some time, and what will happen, it comes down to the baseline. So and till the next dose administration it will be a dose cycle, and B will be the contact time. So B will be the one which will tell you about how much concentration of drug is required to produce this response. So based on this response and comparing with the standard one, you're going to tell at what concentration the unknown compound is producing this effect in comparison with the test compound. So here you see for same thing, you can see the fast contracting tissue as well. So what are the types of tissue in isolated tissue experiment? So according to the tissue or muscles obtained, we have a smooth muscle preparation as well as skeletal muscle. So usually for smooth muscle, we use the uterus, the tracheal smooth muscle, vas deferens aorta, commonly used ileum, ascending or descending colon, etc. Skeletal muscle, we are using a rectus abdominis muscle. So especially the frog rectus abdominis muscle, we can choose for testing the skeletal muscle activity, and frog's art for the cardiac muscle responses. Then we have a, according to the response obtained, you can classify into the slow contracting tissues like frog rectus abdominis, the fundus, chick biventer, cervix muscle, guinea pig tracheal smooth muscles, they belong to slow contracting muscles, whereas the ileum, uterus, ascending colon, descending colon belongs to the fast contracting tissues. So how do we classify the bioassay into? We classify it into direct endpoint assay, which it measure the response of tissue substance at a specific endpoints. So endpoints are kept at what dose it will produce the response we are going to check. That is the direct endpoint assay. And quantal assay, it's about the all or none phenomena, whether this drug produces a convulsion or not, whether this drug produces a hypoglycemia or not. We need to check a quantal assay based on the response being either present or absent in a population. Then we have a graded response assay where we have a multiple sub assays like bracketing assay, where we need to set a standard doses around the test dose. So we need to give a first test dose followed by we need to give we need to give a standard dose first, minimal standard dose, minimal height will be there, followed by a test dose, and it will be bracketed with the larger dose which we choose from the standard drug. So the outer responses will be of the standard drug responses, whereas in between will be the test drug responses. The matching assay is used to compare the test dose within the known standard for similar response. So you're going to increase the drug concentration of the test drug onto the inner organ bath where tissue will be attached. You'll be increasing gradually, and you're going to check at what height the test drug reaches the standard concentration or the standard drug dose. That concentration will be matched. And in interpolation assay, you're going to estimate the potency by comparing the test drug to a reference using the interpolation. So this is requires a lot of little bit data where we need to be monitoring properly, and properly we need to extrapolate, and we need to put it on the graph to compare the potency of the test drug and the standard drug. Then we have multiple point assay where we use several doses, that is 3-point assay, 4-point assay, 6-point, and the 8-point. So when we compare all those drug assays, the multiple point assay is more accurate, it is more reliable when compared to the other types of assays. So coming to the direct endpoint assay, as in brief, basically determine threshold required for response in each of the experimental unit. So basically it is the ratio of these doses which will going to estimate the potency of the test preparation relative to the standard drug. So this is the common thing which we're going to do in all the assays, where we compare the potency of the test preparation in relation to the standard dose. So usually the design which we choose in direct endpoint assay will be the crossover or parallel group design, where infusion of test and standard preparation will be done at fixed rate until a direct effect is observed. So at fixed rate, you're going to infuse both test and standard drugs, and you're going to see at what uh, concentration the effect will be seen at both standard and the test preparation. So example is the when we give a digitalis, the stopping of heart with digitalis in cats. So this is with respect to the example of direct endpoint assay. So this is the standard formula which will be used to calculate the threshold dose of the test, that is the threshold dose of standard into concentration of the standard drug. So what is the advantage of direct endpoint assay? So rapid drug effect can be detected, easy to recognize the responses directly proportional to the drug effect and the dose, because you are infusing the drug at a fixed rate, and quick endpoint detection can be made. You are not doing a serial dilution, constantly you are infusing the drug, and at some concentration you're going to see the response. It is easy to be done and easy to recognize the response as well. So what is the disadvantage? It is limited only for checking the toxicity or high dose studies. To check for any lethal toxicity or higher dose toxicity, you can it is used for that purpose. So not suitable for dose ranging studies. Coming to the quantal assay, or the all or none phenomena, or the all or none assay, where you're going to check whether the response is present or not. For example, when you do a response, for example, you're going to check for the lethal dose 50 determination. So whether you're going to check, example, what we can code as the insulin assay or mice convulsion method, where you're going to see whether the mice will get a convulsion or not. In insulin assay, you're going to check whether the preclinical animals will affected with hypoglycemic attacks or not, etc. So lethal 50 can be determined. Advantage, usually it measures a response where effects are either present or absent, helps to determine the lethal dose. So please remember, quantal assay is used to determine the lethal dose as well as the potency of the drug. Disadvantage, the responses are irreversible. If the death occurs, so it cannot be reversed. So animals are used only once in the quantal assay, because it's a all or none phenomena. So if survives, survives, otherwise there is no. So that's why it is called as all or none assay. So effects are non-permanent. You have to go for multiple tests again with the same species or the same animal as well. Coming to the graded response assay, so usually here you'll get a smooth sigmoid response curve, where you can beautifully see the floor and the ceiling responses at the extremities. So you would have seen a dose response curve where you'll get a sigmoid response in ideal dose response curve. So this is the one which is used to determine the drug potency by comparing the mean responses of the test drug with the standard drug. So basically it uses the analytical dilutional assay. That is what we are doing in the bioassay, where we will be checking for the serial dilution of the test as well as the standard drug. And based on the serial dilution, individually we check for at what concentration, at what dilution these tissues are responding. That is called as analytical dilution assay. So example is the contraction of the smooth muscle preparation for histamine assay, or we can study the BP responses in case of adrenaline. So whether the blood pressure is decreasing or increasing, or smooth muscle is contracting with respect to the histamine assay. So next, coming to the advantages. So it's very simple, effective in terms of determining the potency of the drug, does not require any statistical analysis or any complicated procedures are not required. Since we have done serial dilution properly, if we are doing a dilution at a proper rate and correct values, we'll get a proper response. Disadvantage, the determining the ceiling or maximal effect, minimal response experimentally will be challenging. So again, why this is challenging? Those individual concentration, unless we prepare properly, will not get a response as well. So this is how serial dilution will be made in a descending order. This will be the highest concentration followed by the lowest concentration. So based on that, we need to check the lowest concentration. We can use these concentration initially, then go for the higher concentration to check at what minimal dose the drug, this unknown product will be producing the response in the biological tissue. Later, you can go with the maximum dose and check for the tolerability or the lethal dose or the maximal effective dose and the lethality or the what at what dose the adverse effect will be seen can be seen at the higher doses. So coming to the bracketing assay, again the bracketing assay is used whenever the tissue samples are low. So whenever you calculate if the tissue sample is very small, a small piece of tissue is there. So in such case, you can go with the bracketing assay. So bracketing assay will helps to cut down the time, and only few responses are enough to determine the concentration. So the response is bracketed between two responses of the standard drug. So standard drug you will get initially you will get one lower standard drug response, and later followed by a test drug, and later at the end you will get a higher dose response of the standard drug. So usually potency of the drug can be calculated directly. And again, when we do the bracketing, we need to go for interpolation on the dose response curve, so that we would have checked only the lesser responses or a small number of responses. That's why you need to go for a interpolation. So sample advantage, sample and effective, simple and effective when the test sample is limited, and limitation is poor precision and reliability, because only limited responses we are collecting and we are interpolating that later on the dose response curve. So that is the major problem of bracketing assay, where we have poor precision and reliability and unable to calculate the errors accurately. Only the four responses are calculated in case of bracketing assay. Coming to the matching assay, again the principle is same. We are comparing the potency between the unknown and the standard. It is basically it is matching means just you are using a trial and error method. You are checking for at what dose this will produce a similar response to that of the standard drug response. So response is matched at one dose. Once you eliminate that dose, you need to go for the another dose. So you need to go for dose response curve to test the potency or drug concentration of the test drug. So it requires very small sample volume to test the concentration or potency of the test drug. So what are the advantages? Again, it is simple, does not require any dose response curve. The limitation is it can be a subjective and it is more susceptible for experimental errors unless it is conducted in a controlled manner. So no parallelism in response. There can be error with respect to the standard curve of a drug. So in such cases, to determine the potency will be very difficult. And very important, the tissue which is required when we are conducting a matching assay should be very sensitive. So to make tissue selection, it becomes very crucial, because matching assay, the difficulty is arising at the potency, because the parallel shift of the dose response curve may not happen when we are comparing with the standard and the test drug compound. So coming to the interpolation assay, so again basically it is the dose response curve of the standard drug prepared the concentration of the unknown drug. Again, the graphical representation come into place where will be interpolated on the dose drug response curve graph. Again, we need to quantify and extrapolate. So again, log dose of the linear response relationship will be calculated. Again, it takes a longer time. So advantage is you can quantify and estimate the drug potency. That is the advantage. But limitation is it requires accurate plotting on the graph, and also the assumption of the linear dose response relation should be very accurate. Again, all those comes in the background. What are the standard precautions we are taking in conducting the experiment, preparing physiological salt solution, calibrating the instrument, choosing the tissues as well. So although we can quantify the drug potency, uh, plotting will be very difficult. Coming to the multiple point assay, where you're going to use multiple responses. So this is multiple point assay will address some of the limitations which were there for the other assays, like sensitivity, accuracy, methodological errors can be removed by using the multiple point assay. Since we are taking a graded response assay in case of the multiple point assay, we are minimizing the tissue sensitivity errors, as well as we can improve the methodological accuracy. That means we are increasing the reliability of the assays. So procedure, what we are doing here is the select one or more doses of the test compound, then you compare with the two or response of the standard and focus on the linear portion. That's what. So we need to select the dose from the in between the 25 and 75% of the dose response curve to get a better dose discrimination between the two concentration. So advantage, it improves accuracy, it increases the sensitivity. The limitation is it requires careful dose selection and accurate response recording. So you need to select the dose in from that particular range only, 25 to 75%. If you choose less than 25%, above 75%, the concentration or the tissue may not respond and you will not get a proper recording. So how it can be done? So this is about the bracketing assay. We'll go for the multiple point assay in the subsequent slide. The bracketing assay, what you do is you take standardization. Initially, you standardize the graph. Then you go for a S1, that is the standard S1, and S2 are the standard drugs, and these are a two concentration. Then you have to plot the test drug. Again, you check for the normality or the response. That you standardize the response of the test drug. Once you standardize the response of the test drug, now we are going for the bracketing assay. So what we are doing here, initially we plot S1, S2. Again, these are checked with the comparison with the test drug dose or test drug concentration, comparing with the standard drug. Once you have done the standardization with respect to the test and standard drug, next what we are going is the select the lower dose of the standard drug, select the lower dose of the standard drug, and after that you administer the test drug, and after that you again go for the higher dose from the standard drug. So this is bracketing. So in between the standard, you are administering the test drug to get a response. So that is a test response is bracketed between the two doses of the standard drug S1 and S2. Then he coming to the matching assay. So here again, matching assay, what you need to do, standardize the response with the standardized drug S1 and S2 or the different concentration. Again, you standardize, and later what you need to do is you need to match with the standard drug response. So initially again TS1 is tested for the standardization. So what we next you are doing is the test drug is chosen. You can increase the dose of the test drug in such a way that until it matches with the S1, that is the standard dose response, you're going to increase the dose of the test drug. So in such a way, you're going to match the test drug with the standard drug, and you're going to calculate the test drug concentration. So this is how interpolation bioassay will work. So again, this requires a graphical representation where log is required. You need to calculate the log dose response, etc. And it is a little bit cumbersome, but it will give what we can extrapolate for a larger quantity of drug as well. Here, as we said, for multiple point assay, you need to select the dose from this linear portion of 25 to 75% only to get a better dose discrimination. So otherwise, what will happen? At higher doses, toxicity will occur. There is a ceiling effect. At lower doses, the tissue will not be very much sensitive to respond well to the lower doses. So this dose between 25 to 75% will be ideal to differentiate between the each doses or the concentration. And next we have this is a type of the multiple point assay, that is the 3-point assay. So where we make use of the Latin squared randomization. So 3-point assay means there is a 3 responses. So there is a 2 response from the standard dose and 1 response from the test drug. So similar concept is utilized. 75 we are choosing the dose from the range of 25 to 75% of the maximal response. So how do we get this? This we do it when we are standardizing the graph. So when we standardize it, that from the standardization initially we need to select the dose from 25% to 75%. For example, you are giving a 10 milligram of a drug and 100 milligram of a drug. You need to quantify what responses we are getting. So if you are getting a maximal response and a lower response, you need to make sure that you need to select in between those responses, and procedure, randomize the 3 selected responses for accurate bioassay comparison. Again, we need to estimate the relative potency by comparing the parallel log dose response lines. So advantage, it is more accurate, it is more precision in calculating or estimating the potency, and we can extend the linear range for better dose discrimination. And limitation, it requires that careful dose selection and randomization should be done properly. So how it is done? This is the Latin square randomization. So it will have a 9 responses. 3-point assay will have a 9 responses. We have a standard drug 1, standard drug 2, 2 different concentration, and we have a test drug. And again, you randomize by S2 T S1 T S1 S2. You take an average of it, and this is how you calculate the relative potency. So this is the height will be calculated, and T minus S1 divided by S2 minus S1, and log of S2 by S1. So T, capital T and S1 will be the height of the dose, height of the test dose. S1 will be the height of the standard dose. This S2 will be the height of the standard concentration of S2. So S1, small S2 and S1 will be the concentration of the standard dose, and small t will be the concentration of the test dose. So this is the 3-point assay. First, you will get a DRC of standardized drug, that is S1 and S2. Then DRC of the test drug has to be calculated to standardize it. Following, once you standardize the graph, why we need to standardize? We need to check for whether the tissue is reacting to the standard drug as well as the test drug as well. So once we are okay with the responses, later we can go for a 3-point assay where the 9 responses are done, where in the serial order similar to that of the Latin square, that is S1 S2 T, then followed by S2 T S1, then T S1 and S2, and you take a average value and you calculate the height. In such when we are calculating the height and taking the average or a mean value, we'll be able to minimize the errors. So that's how this 3-point assay or multiple point assay are more accurate in reducing the bias in calculating the potency of the drugs. So this is the formula to calculate the unknown compound. So a number of x times more concentrated than standard, S1 into anti-log of mean of the test compound. So that's what we are talking. S1 and S2 were the length of the standard response. Capital T will be length of the test response. S1 and S2, small S1 and S2 will be the standard dose for S1 and S2 correspondingly. And T will be the test dose, small t will be the test dose for corresponding to the T, that is the standard test drug, and MT will be calculated along mean of these concentration, that is the from standard and test response positions. So how we need to calculate the error? We use a percentage error calculation where we take a actual concentration of the test minus the observed concentration of the test and into and 100. So permissible limit for error when we are calculating the multiple point assay will be less than 10% should be the error. Again, in 4-point assay, what we do is similar to that of the 3-point assay, but we are taking a two doses of standard drug and 2 doses of test drug. In previously in 3-point, we are taking 2 doses of test standard drug and 1 doses of the test drug. But in case of 4-point assay, we are collecting 2 doses and 2 doses of standard drug and test drug respectively. So again, the same thing will be followed. We'll get 16 randomized responses, and it allows statistical validation and confident interval estimation. So since it achieves the parallelism between the DRC, it is very essential in terms of maintaining the parallelism to calculate the accuracy of the drug response. And again, it is more precise, reliable than 3-point assay as well, and it has got better control over the control. It controls the error as well as got higher sensitivity. So that's what whenever we ask about which assay has got the higher sensitivity and more accuracy and lesser bias, your answer should be multiple point assay, and multiple point assay as well. 4-point assay is more superior to the 3-point assay in terms of precision and reliability and sensitivity. So this is how the Latin square for 4-point assay looks like: S1, S2, T1, T2. You go with randomization, you'll get a 16 responses. This is how you calculate the potency of the drug here. S2 minus T2 plus S1 minus T1. This will be the height and this will be the concentration. So later you plot it. This will be the mean what you get. Then you calculate for the concentration of the unknown compound S1 by T1 into anti-log of the mean. So that's how you get a unknown concentration value. So the height has to be calculated graphically. Again, you need to plot on the graph sheet and you need to calculate for the values. And this is the 4-point assay, similar to that of the 3-point assay. What you saw initially, we need to standardize the heights for the standard drug and the test compound. Later, you go with the S1, S2, T1, T2. This will be the 1, and this will be the second, third, and 4. 4-point assay, you take an average of the S1, all average of S1, all average of S2, average of T1, average of T2, height, and you calculate. And also the dose concentration which was used. The dose concentration which we'll be using for S1, S2, T1, T2 will be, that's what you will be selecting from the 25% to 75% range. Then 6-point and 8-point assay are existing now, where we are again, these assays have got higher precision and low variability in terms of estimation of drug potency. So these are again superior to 4-point assay. 6-point assay is more superior to 4-point assay, and again 8-point assay will be more superior to 6-point assay, and it requires very longer period of time and also the patience in conducting these assays. So that's what in 6-point assay, what we'll do is the 3 doses of standard and 3 doses of test will be calculated. Again, in the 8-point assay, what we'll do is the 4 doses of the standard and 4 doses of the test drug will be calculated. So it, as we said, it provides improved accuracy over 3-point and the 4-point assay. So in 6-point, how many responses we are making? We are making 36 responses, whereas 80 in 8-point assay we are making a 64 response. So to get a 1 response, to get that high quality of precision in terms of without having any error, without having any limitations with respect to the tissue viability, the instruments and all, it takes very lot of time. We need to record the responses for 36 times, here 64 times. And again, very important, if it is done properly, we'll get a maximum sensitivity and low experimental errors. So what are the advantage? Again, it has got higher statistical power, higher validity. It will reduce both biological and methodological error, and we can have a detailed dose response curve analysis. The disadvantage will be very time consuming. Probably you need to you need to do for whole 2 or 3 days if you are doing a 64 responses, and resource intensive, and you require a proper tissue preparation and environmental, that is controlled lab preparation should be there. You should have a highly accurate research setting and regulatory submissions as well to conduct such type of 6-point and 8-point essay. And this is a chart which is the gives a easier comparison to you look to revise and analyze what are the essays and what type of response they are checking, what is the number of doses, whether how much single dose has to be given or standard dose, how much has to be given has been recorded here, and what is the sample volume which is required. As we said, for bracketing and matching, only we require very low doses is required. And precision, when we take, excellent will be the 8-point essay, very low will be the direct point essay. Time required, direct essay will give a quick response, whereas the 8-point essay is the very long time consuming experiment. Statistical analysis, very valid and accurate with respect to the 8-point essay, whereas here it is not essential. So that's how this is the summarize. So our purpose of the bioassay was to compare the potency of the unknown compound and the standard drug. Basically, it evaluates the biological variation or the biological responses. So basically, why we need to have this to determine the therapeutic potency or therapeutic equivalence with respect to the standard drug which are available. So we need to come out with the newer drug. So to come out with the newer drug, we need to go for a preclinical bioassay before it comes to the clinical trials. So type of tissue, what were the types of tissue? We had a smooth muscle, skeletal muscle, and cardiac muscle. And examples are the ileum, rectus, abdominis, and frog heart. Then what are the two types of responses we are seeing? Slow contracting and fast contracting. And these are the tissues for the slow contracting and fast contracting tissue responses. For physiological salt solution, commonly we use the thyroids or the frog ringers, ringer lock, and the Krebs, and these are the tissues. To these tissues, these are the specific physiological salt solutions. Then we have two types of transducers, isotonic and isometric. Isotonic will look into change in the length, whereas isometric will look into change in the tone of the muscle, but not the length. So dose of the cycle component, we have three components: baseline component, dose response, and washing. This has to be a standard thing for each of the responses, what we collect after administration of the each test concentration as well as standard concentration with respect to the tissue which we are seeing with. And we have a type of assays. We have a direct endpoint assay, quantal assay, and graded response assay. Apart from that, graded response assay are broken down into bracketing assay, matching assay, interpolation assay, and multiple point assay. And usually when we are talking about 3-point, 4-point, 6-point assay, you are taking a increased number of responses. You take 9, 12, 36, and 64 respectively with the 3-point, 4-point, 6-point, and the 8-point assay. So coming to the end, so bioassay is about comparison of the potency of the test drug with respect to the standard drug. It's a quantitative analysis, so where you quantify the amount of drug which is required to produce the response with respect to the biological tissue. So you are using this bioassay for drug evaluation, for testing, as well as in comparison with the standard drug. You need to evaluate the test or the unknown compound. So for evaluation of the test compound, we are utilizing the bioassay in comparison with the standard drug. And also make sure that there exists a biological variation. So from species to species, the drug response may vary. Even you are choosing the same species may react with variable responses. And therapeutic potency, So basically, at the end, what we need to know is at the end of the bioassay, we need to know what are the comparison of the unknown compound as well as the test compound, unknown and the standard drug potencies. That's how we develop a newer drug. So unless we have some potency, it will be passed on to the next stage, that is the clinical trials. If you are not finding any of the positive results, you're going to discard the study or stop the study there itself, so that we can save a lot of time and the money as well as economy without wasting much resources. So this was all about the bioassay in brief. Thank you. If you have any doubts, you are free to ask me.
Thank you, sir. If you have any doubts, you can type in the chat box so that it can be answered. Yes, the PPT and this recorded session will be shared in the WhatsApp group. Yes, Jai Mahakal and Sumit, you can type your question in the chat box, please.
Sir, bioassay are still used in the industries. That's what, there are automated instruments which will be used to measure the tissue responses, sir. Yes, from endpoint assay till we get, since endpoint assay we get a direct quick response, we will be testing till we get a response. Now, once we get a supramaximal response, we will not be able to get those responses again. So that's why in case of when we go for a multiple point assay, we are choosing from 25 to 75% of the range. So that's what, sir, why we are choosing between the 25 to 75% means above 75%, you will reach a maximal dose where even tissue will not be able to respond. At the lower dose, sometimes tissue will not able to pick such a lower concentration to respond to that concentration. And higher concentration, there can be desensitization of the tissues as well, and it may not respond as well. So that's why we are picking in between mid average range, 25 to 75% range, where tissue will be able to pick the response. Sir, with respect to the bioassay books, you can go with one very interesting book is there, written by M.N. Ghosh. It's about experiment fundamental pharmacology by M.N. Ghosh, as well as one more is there, experimental pharmacology and clinical therapeutics by Dr. Bikash Mehdi. You can refer those two books, sir. M.N. Ghosh and Dr. Bikash Mehdi. So, ma'am, usually in academics, we go for the quantitative bioassay where you will be using, most of the times, we will be using a single organ bath and we'll be doing it. So, since when we came to the post graduation, that time it was the like dilemma of doing animal experiment. Just we had during that times we have tried with the chicken ileum as well, since the dissection of the animals like mice, rats and guinea pigs were banned. So even with respect to the chicken ileum, we'll get a good response when we are testing those with the unknown compound. Yes, for learning purpose, we'll be using the known compounds only, ma'am. So this is whether when we are discovering a newer drug, we'll be going for the unknown compounds. So as of now, as far as much my knowledge is concerned, the bioassay is being used for both smooth muscle as well as the skeletal muscle, sir. Is it ideal to take a lowest possible concentration of the known stock solution initially to check for sensitivity of the tissue? Please start with 100 dilution and do standardization. That's what, ma'am. Even if you go with the 100 dilution or lower doses, initially you need to do the standardization with respect to both the test drug as well as the standard drug. Initially, even if you go for a higher doses to check for the tissue response, once you wash the tissue, again you need to check the tissue viability. You need to go for the lower dilution itself. That's what bracketing assay is done whenever the tissue sample is very low. Matching assay requires a little bit larger sample and it is time consuming. But accuracy wise, matching will be more accurate when compared to the bracketing assay. Since the tissue is small, you are doing bracketing in between the standard dose. You are keeping test response and just you are comparing with the standard. That's right, man. One of the slide I had given for testing the compound, uh, whether we are selecting the smooth muscle or the skeletal muscle, and based on the tissue which we are selecting, and also when you are selecting the tissue as well, you need to know for those tissue which are the receptors, and if you are able to come with those things, we'll be able to figure it out that which are the tissue has to be selected for those concentration. Usually, as we know, rectus abdominis, you can take rectus abdominis of any animal, but in frog, rectus abdominis is very much more sensitive for the response. So that's why we select those rectus abdominis from the frog itself. So based on the receptors and tissue sensitivity, we're going to select the animal tissues. Since nowadays the AI is coming into the practice, so nowadays even the bioassay has been, as you said, the simulation studies have been come into place. Uh, this lecture sufficient for endopharmacology final exams, Viva, OC, and yes, ma'am. This will be basics of things about the plans and all. Little bit you need to go for the literature search, and you have to read individually more about each of these essays. This will be sufficient. Almost 60 to 70% it will cover your MD pharmacology exams. So what standard we do in fixing the solution means usually we shellac can be used with methanol. If shellac is not there, even methanol can be used for fixing the chymograph graph, ma'am. And nowadays, since digital this has come nowadays, digital chymograph recording has come where it can record on the computer software. We no need of smoked chymograph sheets. Yes, sir. It can be used now. As I said, we have got a digital this thing where the digital chymogram will reflect those graphs in the computer itself. So that can be downloaded and that can be measured. So basically, from the pharmacology exam point of view, what you know is you need to understand the definition of the bioassay. What are the types of bioassay? What are the advantage? What are the application of bioassay, and how these individual bioassays are done? And they may ask about the what are the parts of the components of the bioassay, like organ bath, the chymogram, and how the magnification will be done, how it is calculated, and how the dose responses will be selected, what are the drugs which will be used for getting a response, what are the tissues which you are going to select, and which drugs are more sensitive for which tissues, and what are the types or the methodology you follow with respect to the types, how bracketing essay is done, how the matching essay is done, all will be asked in your exams. At least you should have a basic knowledge of what is done in the bioassay, how these types of bioassay are conducted, and for what purpose. So if you have any doubts, you can leave your doubts subsequently also. So we'll be able to communicate.
Yes, hope I think all the doubts have been cleared. Thank you, sir. And it was very fantastic class, and it was very crisp, and you made it very clear, understanding bioassay. And in case if you all have any doubts, you can contact me so that I can share the questions with you, sir, so it can be answered. Is it fine, guys? Give a thumbs up if you all okay. Yes, yes. This video and even this presentation will be shared in the group. Okay. Thank you, sir. We can. I think we can wind it.
Okay, sir. Thank you. Thank you all the participants. Thank you all.
Thank you, everyone. Have a great day. And all of your certificates will be provided within a week. In the link will be provided to your mail ID, and also in the group also it will be provided. Thank you, guys. Have a great day.